Now I'm thrilled to introduce today's Huddle speaker, Sujit Pujhari. Sujit is an assistant research professor in the Department of Entomology and the Center for Infectious Diseases Dynamics. He earned his Ph.D. in medical virology from the Postgraduate Institute of Medical Education and Research in India in 2011. He also received training in vaccine designing and efficacy study from the Vaccine and Infectious Disease Organization International Vaccine Center at the University of Saskatchewan in Canada. And I could tell that you're all impressed that I can pronounce Saskatchewan. Sujit joined the Department of Entomology at Penn State in 2015, with his research focusing on multidisciplinary approach to the study of virus life cycles, virus structure, and their interaction with human host and mosquito vector. He is also working on developing a novel preventative and diagnostic strategies for emerging and re-emerging mosquito-borne viral infections. Please help me welcome today's Huddle expert, Sujit Pujhari.
Thank you, Paul, for a nice introduction. It's my pleasure to be here this morning and share my work and effort on my dream of a world without infectious disease, a world with less suffering, a world with less hardship, and a world with less distress, at least from the problem of mosquito-borne viral infections. From last 13 years, I'm working on mosquito-borne viruses from different perspectives, ranging from human host, mosquito vectors, and pathogen itself, in order to get a novel insight into the biological process that drive the biology and evolution of infectious agent, and process that can be used to limit their harm to the individual and population health. Before starting the discussion on the problem of mosquito-borne disease, let's understand the magnitude of the problem of infectious disease as a whole. Infectious disease does not discriminate among gender, age, and race. Even does not have any geographical boundary. It affects people all around the globe. According to World Health Organization, in a real-time scenario, 1 out of 6 individuals around the globe are affected by infectious disease, and 32% die every minute. What does that mean? In next 60 minutes, while we are discussing the problem of infectious disease in this closed room, 1,920 people will lose their battle against infectious disease. I know some of you might have an understanding about infectious disease, but for those who are new to infectious disease are disease caused by microorganisms that invade tissue. They are caused by microorganisms like virus, parasite, bacteria, and fungi, including yeast. They need certain kind of vector for their transmission. That includes contaminated water, contaminated food, animals, insect, including mosquitoes. More than 70% of emerging and re-emerging viral infections are vector-borne or zoonotic in nature. The spread of West Nile virus and the recent arrival of Zika and the rapid spread around the world, the outbreak of chikungunya virus in Indian Ocean and in different parts of the, like in Europe, and continuous increase in the number of cases in dengue shows the danger posed by mosquito-borne viral infection that has spread beyond their traditional geographical boundaries. Many of you surprised to know that mosquitoes are one of the deadliest animals on the planet. They are responsible for death of almost close to 1 million people every year by transmitting and infecting viral, parasitic, and nematode infection. Well, there are almost more than 3,500 species of mosquitoes around the globe. And 176 species are found in the United States. And here at Pennsylvania, we have around 62 species of mosquitoes. Many of them can successfully carry human pathogens. But there are only few mosquitoes which can successfully carry and transmit those pathogens to human. Mainly, Aedes Culex, and the Anopheles group of mosquito can transmit human pathogens. Okay, one of the graph is missing from here. Sorry for that. But anyway, what I want to bring your attention is that the Aedes aegypti and Aedes albopictus are the main vector for many of the viral pathogens. They have successfully, like during the process of evolution, they have acquired the character of get established in a new geographical location. Previously, they were located to the African subcontinent, but now they are everywhere. They have amazing power of adaptation to the new world. And they are responsible for transmission of most of the viral infection you heard of. They are responsible for transmission of yellow fever virus, Zika virus, dengue virus, chikungunya, and even Mayaro. This graph represents the geographical distribution of the Culex group of mosquitoes, and they are mainly responsible for transmission of West Nile and Japanese encephalitis. The one extreme on your right is the distribution pattern of Anopheles group of mosquitoes. They are also known as malaria mosquitoes. Beside transmitting malaria, they are also responsible for transmission of certain viral infections like Wanyangnyang virus and Mayaro virus. In nature, basically those vector-borne diseases are maintained in the forest by forest-dwelling mosquitoes, forest-dwelling non-human primates, mammals, and birds. Occasional spillover happens when human beings come in contact with their natural cycle. And because of urbanization and rapid expansion of global population, the spillover has become a more frequent event. Once you get bitten by an infected mosquito, the infected mosquito releases some of the infected viral particle under your skin, where the virus replicate, and then find some local lymph node. From the local lymph node, it makes their way to your bloodstream. And this time period, like from getting bitten by infected mosquito to release of the viral particle in your bloodstream, this take roughly 4 to 14 days, depending on the virus. And this time period, what we call the incubation time period. And during this time period, you are perfectly healthy. You do not show any symptom. But after the incubation period, you may develop some mild flu-like symptom. That may include fever, headache, may develop rash. You may have joint pain, muscle pain, or redness of eye, what we also call like conjunctivitis. If you are lucky enough, that's all. Within 7 days or 7 to 8 days, you are back to your normal routine. You are back to your office. But everyone is not lucky enough. Depending on the type of viral infection, they may end up with severe form of disease. If the virus is neurotropic, for example, West Nile virus, equine encephalitis virus, Zika virus, the virus make their way to your central nervous system. It infect your central nervous system, and the patient develop with certain neurological complications. Virus like chikungunya and its related virus, they make their way to your joints, where they replicate and develop autoimmune-like disorder. And the patient end up with debilitating joint pain, which can last to a few weeks, but in some cases, it could last up to a couple of years. Liver is one of the vital organ of your body. It detoxifies most of the toxin your body produces during the process of metabolism. Yellow fever virus has a tropism towards your liver, where it infects the liver cell and destroys some of those liver cells, and the patient develops jaundice-like symptom, which could be fatal in nature. Not only that, very recently, Zika virus has gained a function to cross the placenta. If a pregnant woman gets bitten by a Zika virus-infected mosquito, the virus can cross the placenta and infect the developing baby inside their womb. Some people lose this severe form of the disease. Ultimately, they die. And the case fatality rate varies from virus to virus and outbreak to outbreak. But there are some brave individuals who survive the even severe form of the disease. If a person, for example, recover from a neurotropic viral infection, they may develop poliomyelitis-like symptom. They may develop learning disability. They may have speech impairment. And in case of Zika virus infection, the baby born with a small head. And he or she has to live the rest of their life with multidimensional disability. Diagnosis, prevention, and treatment are the key to fight infectious diseases. Especially in case of viral infection. The viral infection, they have a basically close association, or they use the host machinery for their life cycle that make things more complicated. Because of that host-dependent life cycle, it's very difficult for us to develop an antiviral agent for those viruses. It's difficult for us to come up with a new vaccine strategy or to come up with vaccine candidates. So not only the viral host-dependent viral life cycle, but also the size of the virus also bring a lot of challenges for us. To give you an idea how small those viruses are. Compared to a salt grain, it's almost like 80,000 times smaller than the salt grain. Compared to your tip of your hair, it is almost tip of your hair. It is almost 400,000 times smaller than that. Yep. And we need high-resolution microscope to see those viruses, to understand their life cycle. Each and every laboratory in the world cannot have those high-end microscope in the lab to understand the viral infection. But in the meantime, we have developed certain kind of strategies to understand those life cycle of the viruses. What we did is we use different kind of fluorescent tag, where we are able to tag those viral protein on the viral surface. Also, we developed certain tag that can identify the viral genome. And we use the fluorescent technology to understand the biology of the viruses. This is what you are seeing is a human cell tagged with red dye and a blue dye and focused under a fluorescent microscope. The red stripe, what you are seeing, these are called cytoskeletal protein. They provide structural integrity. They give the shape to a particular cell. And the roughly oval or circular blue structure, what you are seeing, those are called the nucleus. I infected these cells with a mosquito-borne virus and tagged their genome with a green dye. And what you are seeing here, the green dots, these are all the genome of that particular virus. This is what you are seeing is a three-dimensional reconstruction of a viral infected cell. And this particular cell, what you are seeing, is full with infectious viral particle. And the viruses are ready to come out from that cell. What I want to tell you is that these are the different, indirect way of understanding the viral pathogenesis or the viral life cycle in the absence of the high-resolution electron microscope. But here at Penn State, we are lucky. We have all those facilities here. This is a viral infected cell focused under electron microscope. The black circular dot, what you are seeing on the outer surface of the lining, those are the viral particles. They are entering into the cell. The lining, what you are seeing, the dark line, this is what we call as the plasma membrane. This is the barrier the virus has to cross to infect a human being or any organism. And this barrier, or what we call as the plasma membrane, this plasma membrane has different kind of protein sitting on that. And some of the protein, the virus use it as a receptor molecule. They goes and bind on those protein. And after this interaction, the virus induces certain kind of signaling event, some event in the cell, so that you can see here, and using that signaling event, they produce those cup-like structure. And the virus enter into the cell. Once they are inside the cytoplasm, the virus make their way, they release their genome into this space, what we call at cytoplasm, where the viral genome get replicated, and later stage, from the genome, it produces a lot of protein. And then they assemble into immature viral particle. And then again, they come back to the plasma membrane, from where they take out a little bit of your plasma membrane, and the fully infectious viral particle, they come out. And these infectious viral particles are again ready to infect a closed by cell, or they are ready to release into the bloodstream. So when the next time when a mosquito come and bite you for their blood meal, they will take those infectious viral particle along with that the blood meal. And the virus get established in that. The infection get established in the mosquito. And when the next time the virus goes and bite to a healthy individual, they release some of those infectious viral particles and make the person infected. As I told you, here at Penn State, we also have the super resolution or the cryo-electron microscope. Using this technique, we purify the viral particle and take a picture of the viral particle in different dimensions. And then finally, we reconstruct the three-dimensional structure of the virus. And this kind of structural information provide more information about how the virus how the different on the surface of the virus are arranged, how this protein interact with the host receptor. And these information are very crucial for developing vaccine candidate, developing therapeutic antibodies, developing antivirals. And recently, we identified a protein called HSP70 on the cell surface, which is a co-receptor for Zika virus. This particular protein also helps in the different other stages of life cycle of Zika virus. For those who do not know no idea about what is a heat shock protein, to give you an idea, when our body temperature goes up, it could be because of a fever, or could be when you are like outside in a hot summer day, your body temperature goes up. Or maybe when you are taking a hot bath in your bathtub, your body temperature goes up. There are some protein in our body which are thermolabile. They do not like high temperature. They start degrading. But those proteins are very important for our body. So in order to minimize that damage, our body produces heat shock 70 protein. And this protein help those thermolabile protein to get stabilized in the heat condition when your body temperature goes up. So coming back to the Zika virus infection, Zika virus take advantage of this particular protein to get into the cell to produce more viral infectious particle. So from this study, what I conclude is, if we the inhibitor for HSP70 can be developed as an antiviral candidate for Zika virus. Now again, switching gear a little bit to human system. So when, as I told you, after an infectious or a virus-infected mosquito bite you, they release some of those infectious viral particles, and ultimately that come to your bloodstream. Mainly in your blood, there are two different kinds of cells. There are some other, but mainly there are two. One is called your red blood cell, another is called white blood cells. The red blood cells are very important for transportation of oxygen to different body. On the other hand, white blood cells are part of your immune system. They are also called as leukocytes. There are different type of, white blood cell. They are very important for your immune system. And these white blood cells are very specialized. The group of cells what you are seeing on your right, those are specialized for bacterial infection, fungal infection, parasitic infection. But the cell what you are seeing on your left-hand side, these are specialized for viral infection. These are called monocytes and lymphocytes. So once your body get infected by those virus, then monocyte see, and if in case the monocyte during the surveillance process, it see one of the the virus, then they engulf those virus. Then they break the viral protein into the smaller pieces, and display those viral protein on their surface and give signal to other immune cell. And in this process, our immune system develop, and they produce different kind of immune system. In one arm, what they do, they just try to find out where are the infected cell, and they destroy that. And the other side, our body produce certain kind of proteins, or this is what we call like either cytokine or antibodies. What the antibody does, the antibody goes and bind on the surface of bind to the surface protein of the virus and inactivate them. So basically, our body produce two different arms of the immune system, what we call like Th1 or Th2. The Th1 is, they basically clear those infected cells, and using the Th2 kind of immune response, they clear the freely moving viral particle in your bloodstream. So a perfect balance of this Th1 and Th2 immune response is very, very important. I'll give you an example how, what happens if there is an imbalance in Th1 or Th2. In case of Japanese encephalitis virus infection, which is a sister virus of West Nile virus, if there is activation of Th1 kind of immune response happen, then the patient end up with more severe form of the disease. But on the other hand, if the body switch to develop a Th2 kind of immune response, the person get clear of the virus within 6 to 7 days. So what I mean to say that the immune system can be your double-edged sword. At one side, it protects your body. At the same time, if the wrong arm of the immune system get activated, then it can harm your body. I'll give you a couple of examples of that. In case of dengue virus infection, I believe most of you heard about dengue virus. But maybe you are not aware that dengue virus is not only one virus. It has 4 different viruses. Dengue 1, dengue 2, dengue 3, and dengue 4. Suppose you are get bitten by a dengue 1, a mosquito infected with dengue 1 virus, and it did infect you. And your body developed Th2 kind of immune response. It has an antibody against the dengue 1. Within 6 to 7 days, you are fine. And again, you are suppose get bitten by the same dengue, a mosquito infected with the dengue 1 virus. In certain after a year or 2 years or 5 years, then your body remember. Your body already had the antibody and it protect you. But things are very different if you are get bitten by a mosquito infected with the dengue 2, 3, or 4. In this case, the antibody against dengue 1 will not protect you against dengue 2, 3, or 4. But at the same time, what it does, it enhances the process of infection. And the patient may develop severe kind of the disease, and they manifest maybe hemorrhagic syndrome, where your body starts bleeding from different parts. It could be from eye, from your nose, from your ear, or from your skin, and which could be fatal in nature. I'll give you another example of, again, the Th1 immune system. How Th1, activation of Th1 could be detrimental. The picture what you are seeing here is the MRI of human brain. The picture on your left is MRI picture of a normal human being. And here you can see a specific pattern of white and gray color. The white color is because of the water molecule inside your brain. Because there is a continuous flow of cerebrospinal fluid into our brain, which provide oxygen, other nutrient, and glucose molecule for the proper functioning of the brain. But in case of Japanese encephalitis infection, the picture what you are seeing on your left, this is a brain MRI of a 14-year-old boy infected with Japanese encephalitis. And here you see this particular pattern has distorted there. This particular virus is neurotropic and goes to a specific area of your brain. And because of that infection, that inflammation happened there. Inflammation is always related to accumulation of water. And that's why you can see this particular area are white. And this specific pattern has gone. And during this process of inflammation, our body also sends some kind of cytotoxic cell to clear those viral infected cells. But our brain cell has limited power of regeneration. While body thinks that he is clearing the viral infected cell, but at the same time it is harming our brain system, like the central nervous system. There are very few lucky individuals survive this kind of infection. And even if they survive, they end up with many disability through the rest of their life. Not only that, I also analyze the blood sample of those patients who survive from the disease and those who died during the process of JV infection. And what I found there is there are certain marker, like in case, for here you can see that there is a significant increase in the concentration of TNF alpha, that's a cytokine, and also significant increase in the amount of interleukin 6. These are the two different cytokines I found highly raised in the person who had or the non-survivor patient in Japanese encephalitis infection. And this kind of information can be used as a biomarker. If we provide this kind of information to the clinician, they may come up with better therapeutic strategies. Or they can manage the patient better. Again, coming back to the infection process, as I told you, even if you get bitten by infected mosquito, not every person shows the symptom. Only 10 to 20% they come up with certain kind of clinical symptoms. That starts with the fever. And again, out of the only few individuals will go to severe form of the disease. Either they may fully recover from that severe form of the disease, or they recover with certain kind of disability, or they never recover from the disease. Why is this happening? It could be because of the viral strain, or it could be because of our immune system, how our immune system is get activated. In case of childhood, our immune system is still developing. When we grow older, our immune system is getting weakened. And even if we are young enough and our immune system is working perfectly, even though we look very similar and we have a very similar genetic code, but still, there is a lot of heterogeneity is there on the genetic makeup. So in order to study that, how genetically we are different, or especially in case of that study group on Japanese encephalitis patient, I look for specific signature on the TNF alpha gene, on the cytokine which is elevated during the JV infection, or in that patient when there is a high level of TNF alpha, those who are dying. And what I found out that the patient which has a very high level of TNF alpha, or those who are dying, they have a very specific kind of signature compared to the other group of patients who survived that severe form of the disease. These kind of information are very, very important when we are trying to develop what we call as more personalized medicine. Having this kind of information is very important when we design or come up with new vaccine efficacy studies. But the drug discovery and the vaccine development process takes time. It takes a lot of money. We cannot wait that long. We need to act fast. We need to protect our people around us. In the process of developing this thing, what we can do is we can target the vector. We need to control the mosquitoes. And we have successfully done it in past. We have eradicated malaria. We have eradicated yellow fever from the United States. So we have a success story behind us. And we did it by using insecticide. We're using a mosquito net, by using biological control like release of mosquito larvae eating fish. But all these solutions have their own limitations. We cannot release those mosquito larvae eating fish everywhere, because there are some mosquito species, they can grow in very tiny amount of water. We cannot reach all their habitat. There are some mosquito species, they bite during the daytime. We cannot control those things using mosquito net. insecticide is also not a permanent solution, because now we are getting some mosquito species which are developing, like getting insecticide resistance. So what we are doing now, we are coming with more innovative strategies to fight against infectious diseases. We are exploring the microbiome of insect. So if you are new to the microbiome thing, you will be surprised to know that our body is inhabited by more than 3 billion of microorganisms, which are very helpful to us. Without them, we cannot survive. So as the insect, they have their own microbiome. Wolbachia is another example, or a part of their insect microbiome. These are a group of bacteria, intracellular bacteria, gram-negative bacteria present inside those insects. And almost 60% of the insect have Wolbachia in them. But what is surprising is that the Aedes aegypti mosquito, which basically transmit most of the human pathogens, they do not have this bacteria in their body. So what we did is we isolated those Wolbachia from different insect species, amplify them in a cell culture system, and then what we saw is that those mosquitoes who were infected or transplanted with Wolbachia, there is a significant reduction in the pathogen load in those mosquitoes. And those mosquitoes basically lose the capacity to transmit the viral infection. And it is true for dengue, chikungunya, Zika, and Onyongyang virus, even for myrovirus. So those mosquitoes who are like when we transplant them with Wolbachia, they cannot transmit the disease anymore. And this is how we generate the Wolbachia-infected mosquito in our lab. As I previously told you, we isolate those Wolbachia from different insects, amplify them in our cell, and inject in the mosquito eggs. And if we are successful, these mosquitoes are successful in hatching those eggs successfully, then those hatched mosquitoes, they are Wolbachia infected, and they carry the Wolbachia in the rest of the generation. And there are some more interesting facts about Wolbachia. Suppose a Wolbachia-infected male mate with a Wolbachia uninfected female and produce some eggs. Those eggs they do not hatch anymore. And the other interesting fact, as I already told you, that if a mosquito is already infected with Wolbachia, they cannot transmit the disease. So we are exploring this interesting fact. And when we are also releasing this mosquito at different geographical locations. And after a few generations of the population is replaced when we release the Wolbachia-infected mosquito, and after a few generations, the mosquito population is replaced with Wolbachia-infected mosquito. This is what World Mosquito Program is doing. They are releasing Wolbachia-infected mosquito in different parts of the world in order to control the mosquito and viral infection. In a recent update, in the last week in Washington, D.C., there was a scientific meeting, American Society of Tropical Medicine and Hygiene, where they reported that there is a almost 90% reduction in the dengue cases in Australia, and almost 60 to 70% reduction in dengue cases in Brazil, where they release these Wolbachia-infected mosquitoes. And this is how this works. This is what we call as like mosquito population replacement strategy. The black color mosquito, what you are seeing, these are disease transmission competent mosquitoes, or the wild-type mosquitoes. So in this kind of experiment, what we do is basically we release Wolbachia-infected mosquito at certain number at different time interval. And after some time, these disease transmission mosquito are replaced by Wolbachia-infected mosquito, which are disease transmission incompetent mosquitoes. And in this way, even if you get bitten by a mosquito, you will not get the disease. Previously, I already told you that when a Wolbachia-infected male mosquito mate with a Wolbachia uninfected female mosquito, and the eggs they lay, they cannot hatch anymore. This is another way to reduce a mosquito population. Especially this is very helpful in case of those invasive species. And this particular method, what we call a mosquito population suppression strategy. This is another article recently published in Nature Journal. This is, I would like to quote you. In this work, Dr. Jiang and his group, they selected some control sites in China, some islands, and they released those Wolbachia-infected male mosquito. And they were successfully able to reduce that particular species of mosquito up to 100%. And similar thing is happening in California too. Because their Aedes aegypti is an invasive species to that particular geographical location. And Aedes aegypti are responsible for transmission of most of the viral infection. So what they are trying to reduce the number of Aedes aegypti population. We cannot win, having said that, I like to emphasize here that we cannot win a battle with a single arsenal. We need to come up with different strategies. That's why we are also working on developing transgenic mosquitoes. We are translating our understanding of viral pathway, viral life cycle, what we understand in our laboratory using the cell culture system. We are identifying the protein molecules or the receptor molecules, what is very important for the entry and the life cycle of the virus. And we are taking that understanding into the mosquito, and we are knocking out those genes, or we are cutting those genes in that mosquito so that we can generate a transgenic mosquito. Those are refractory to those viral infection. They cannot transmit that virus anymore, or they cannot harbor that virus anymore. And this is how we do. We just take those CRISPR-Cas9 that many of you must have heard about that technology. The recent advancement in the genome editing, the precise genome editing. You can just target a specific part of the gene. So we are taking that CRISPR-Cas9 protein and certain the other molecules that what is needed for the gene editing, and injecting that into the mosquito egg. And that's how we are generating those transgenic mosquitoes. But again, this particular technique has a lot of limitation. We need like very high cost instrument. It is very, very labor intensive, and the success rate is very low. So we need to come up with new innovative strategies so that the rest of the world can use their innovative way to control the infectious disease or to make more transgenic mosquitoes to understand the disease biology. And I believe that most of those problems, there is a solution in the nature. And we found a solution in the egg yolk of fruit flies. And here I would like to mention that with a coherent and coordinated and collaborative effort, we all work together in a single unit and turn this idea into reality. And this is how it worked. During the process of egg development, basically the egg accumulates the yolk protein. So we trick that process. So during the process of ovulation and during the egg development process, what the fruit fly does, and so as the mosquito, that they develop a lot of yolk protein. And that is what we call as the precursor yolk protein. And those precursor egg protein are basically internalized by a specific kind of molecule on the developing egg, what we call as the receptor molecule. So those receptor molecule identify those precursor egg protein and internalize them into the egg. So what we did is cross-linked the CRISPR-Cas9 protein into those egg yolk protein. So during the process of accumulation of those precursor egg yolk protein, they take those genome editing material into their egg. And here I also like to mention that when we injected this fruit fly precursor egg yolk protein, the mosquito also take those protein into them. And all the process doesn't done here. We come up with more challenges because we need this CRISPR-Cas9 or the genome editing material to come to the cytoplasm or to outside of that vesicle. And ultimately, it needs to go to the nucleus where it does precise genome editing. So we come up with some more innovative strategy to release that genome editing material into the cytoplasm. And this CRISPR-Cas9, they have a certain nuclear signal that carry all the material into the nucleus where the genome editing happens. So using this technology, what we did was from a very highly specialized microscope and other expensive material. Now you can do this technology by using just $1 mouth pipetter and 10 minutes of training. Because injecting a mosquito egg, you can imagine if the mosquitoes are so tiny, how small their egg could be. It is very challenging. You need a lot of instrument. But here, injecting an adult female mosquito is not that hard. And for this particular experiment, we chose a phenotypic marker so you can see whether your experiment is working or not. And here we targeted the gene which basically is responsible for black eye of mosquito. So using our technology, we just edited the gene responsible for the black eye, and those black eye mosquito turn into white eye. And this is basically a demonstration of our technology is working. So using this technology, or now this technology is available to all over the world, and now the innovative scientists can do their experiment, can come up with more innovative ideas to control the viral infection. And having said that, I understand. And a lot of discussion are still going on, what to do with those transgenic mosquito. Can we release them into the field? I believe right now we may not be at the right stage. We need more discussion. We need to study more on the biology of those transgenic mosquitoes. We do not know what's going to be the ecological impact of release of those transgenic mosquitoes and what could be the other undesired effects. But having said that, what I can say is that using of insecticide is also not that safe, because it affecting many mosquitoes and other insect species. It also has a negative environmental impact and also on the human health. And also we are getting those insecticide resistant mosquitoes too. So we need to come up with a more balanced strategy in future. And with that, what I'm going to say, there is no magic bullet to fight against the infectious disease. We need to come up with more innovative strategies and multidimensional strategies. In the meantime, what we are working on is developing more rapid and sensitive diagnostic tests, designing more vaccine and antiviral candidates to fight against the infectious disease. But in the meantime, what you as a citizen or me as an individual, what we can do? What we can do is basically eliminating the mosquito breeding area around your home, around your neighbor. This could be this is an effective way of reducing the mosquito-borne viral infection, at least in the current scenario. With that, I would like to acknowledge specifically to Professor Jason Rasgon and his team, and also I'm part of that research team. And I also like to thank my collaborator at PSU, and also my colleague in Canada and in India. With that, I would like to thank everyone for being present and listening to this interesting talk. And if you have any questions, I would like to take those questions. Thank you.
Do you have any questions? Right up front.
So is there any truth to the rumor that some people are more prone to being bitten by mosquitoes and other people are far less prone to being bitten by mosquitoes? And can that be exploited in any way to control this kind of stuff?
Yes, it is true that some people are more attracted by certain by mosquito biting habit, because our body produces during the process of metabolism, it produces different kind of chemicals. Even we also produce carbon dioxide throughout our body. So mosquitoes get attracted to certain people because they produce certain kind of chemical what the mosquito likes very much. And yes, we can explore certain kind of those things. And I was reading an article recently, maybe a couple of weeks back, and people are working on that to identifying those molecules and using that particular scent to reduce the mosquito biting habit like thing. Yeah, that's a great question though.
First of all, thank you very much for a very elucidating lecture. I may have missed this in the first 5 minutes, but is there any research being done in terms of trying to increase the natural predators of the mosquito as a way of mosquito control, like birds, et cetera?
Yes. So there are many studies are going on that, and you'll be surprising to know that like dragonfly are like one of the very interesting biological control. Because they in a healthy adult dragonfly can eat almost 100 mosquito per day. So if we somehow can increase the population of dragonfly, we can reduce the population of mosquito in that particular area. And it is being like currently people are doing at different part of the globe. Yes, that's another way of controlling mosquitoes. Yes.
Do you see the application of any of your research or technology possibly being applied to tick-borne infections and diseases that are passed on through ticks?
Yes, especially the work on the CRISPR-Cas9, what we call as a remote control, where we are exploiting the egg precursor protein of Drosophila. And there are many collaborators around working with us, and they have successfully used that technology to knock down some of those genes in ticks. Yeah, definitely whatever we are doing is translatable to other insects too. Yes, please.
Are there any foods or is there any type of liquid that a person can drink or eat that makes the person's body less attractive to the mosquito? Somehow I heard something about research in that direction, and I'd also like to know where the research money is coming from.
Yeah, that's a great question and a nice thought. Yeah, definitely we can manipulate our body metabolism by what we are eating. Specifically, your question, I don't know that which food we can eat, but that's not my area of expertise because I'm a virologist. I'm more focused on like study the virus. So, but definitely, yeah, it can be done. And as you said, like there are some people are doing on that, so I'm not following that. Maybe yeah, that's a good way to control the mosquito-borne virus. And coming back to your next question, like who is funding this kind of research? Like there are many government agency and non-government agency, like coming to listing few of the government agencies from National Institute of Health and National Science Foundation. They are also funding most of the mosquito-borne research. And also there are like many other non-government agency and foundation. They support many of the research on mosquito-borne viral infection, especially Melinda Gates Foundation.
Any other questions? Great. And please join me in thanking.
Thank you.